Pratima and welcome to Planet Physiology. Before we dive into the today's topic, let's start with a quick question. How do we breathe? Is it through positive pressure breathing or negative pressure breathing? And what do these terms mean? Well, normally we breathe by negative pressure breathing. This means our respiratory muscles contract, lowering the pleural and the alveolar pressures, which draws air into the lungs. This is inspiration. When the muscles relax, the lungs recoil and air is pushed out. This is expiration. This entire process happens automatically and rhythmically. But have you ever wondered how, despite being skeletal muscles, the respiratory muscles contract rhythmically without conscious efforts? Pause the video for a moment and think about it. Yes, our automatic breathing is controlled by specialized respiratory centers in the brainstem, and that's exactly what we are going to explore today. We shall first classify the respiratory centers and then discuss their locations, working mechanisms, and clinical conditions related to these centers. This is an important topic in respiratory physiology and often appears as short note, or it is an integral part of essay question on neural regulation of respiration. So let's get started. Respiratory centers are the group of neurons located bilaterally in medulla and pons. They coordinate the activity of respiratory muscles to regulate rate, depth and rhythm of respiration, which ensures optimal gas exchange at rest as well as with changing demands of the body. Respiratory centers are divided into medullary centers and pontine centers. Medullary centers include dorsal respiratory group of neurons, ventral respiratory group of neurons and Pre-Bötzinger complex, whereas pontine centers include apneustic center and pneumotaxic center. So let's discuss each of them in detail, starting with dorsal respiratory group of neurons, abbreviated as DRG. It is called as dorsal respiratory group due to its dorsomedial location in the medulla. So DRG is located in dorsomedial medulla in and around nucleus tractus solitarius. It is represented in blue color in this diagram. Basically, DRG is an inspiratory center and contains I neurons. I for inspiration. These neurons are active only during inspiration. Earlier it was thought that the neurons in DRG initiate basic rhythm of respiration or ramp signal, which then stimulates phrenic nerve to activate diaphragm. But later it was discovered that a specialized region in medulla called as Pre-Bötzinger complex generates the ramp signals responsible for rhythmic contractions of inspiratory muscles. So details of this center we shall study later. Nucleus tractus solitarius is basically the sensory termination of vagal and glossopharyngeal nerves. Hence, it receives signals from variety of receptors like baroreceptors, chemoreceptors and receptors in lungs. It also receives inputs from proprioceptors, central chemoreceptors, deglutition center, pontine respiratory centers and hypothalamus. These signals help DRG to modify rate and depth of respiration as per demand. Now let's study about ventral respiratory group of neurons, abbreviated as VRG. So VRG is located in ventrolateral medulla near nucleus retroambiguous and paraambiguous. As you can note, VRG extends almost the entire length of the medulla. VRG has both inspiratory neurons as well as expiratory neurons, and these neurons are inactive during quiet breathing. But they are activated by DRG during deep, forceful breathing, say as during exercise. Let's see how VRG works. Expiratory neurons in VRG are located in its rostral and caudal regions, and inspiratory neurons occupy the central region. In simple words, inspiratory neurons are sandwiched between expiratory neurons. Inspiratory and expiratory neurons show reciprocal connections where activation of one leads to inhibition of other and vice versa. Rostral part of VRG containing E neurons is called as Bötzinger complex. It sends signals to caudal respiratory neurons, which then send signals to the spinal motor neurons, which innervate muscles of expiration. By the way, do you know why this complex is named as Bötzinger complex? As usual, we think it's a name of discoverer, isn't it? But it has an interesting story behind. You can find it out and share in the comments below. Okay, coming to the last medullary center, the Pre-Bötzinger complex. As shown in the cross section of the medulla, Pre-Bötzinger complex is situated between nucleus ambiguous and lateral reticular nucleus. It contains pacemaker neurons that exhibit spontaneous burst of activity leading to rhythmic breathing. Hence, it is also called as central pattern generator. This picture shows rhythmic discharge from the neurons in Pre-Bötzinger complex of neonatal rat brain slice in experimental setup. In vivo activity is also similar, where rate of discharge gradually increases for 2 seconds and then stops for 3 seconds. This gradual increase in the activity appears like ramp and hence called as ramp signal, and it can be represented diagrammatically like this. So gradual progressive inspiratory signals are indicated by red color, which are responsible for inspiration, and then the stoppage of the neurons causing expiration. So these gradual or progressive inspiratory signals help in smooth progression of inspiration instead of gasping type of respiration. Pre-Bötzinger complex receives signals from DRG as well as VRG to modulate the respiration. Pre-Bötzinger complex also gives signals to hypoglossal nucleus to regulate airway resistance during respiration. Now the next two slides are from nice to know section, and if you are not interested, you can skip this part. The exact mechanism of generation of ramp signals is not known, but it is hypothesized. It is found that rhythm generating core has glutaminergic neurons which generate rhythm and transmit the signals to the premotor neurons within the complex. These neurons in turn lead to motor output to the muscles of inspiration. Core also has inhibitory, either glycinergic or GABAminergic interneurons. Signals from nucleus tractus solitarius stimulates them to terminate the inspiration, which helps to maintain respiratory rate. These interneurons also modulate the amplitude and frequency of inspiratory ramp signal. In their absence, respiratory rhythm becomes slow and stiff. It has been shown that pacemaker neurons express various receptors. The first is NK1 receptors. NK stands for neurokinin. So neurokinin 1 receptors. Substance P acts on these NK1 receptors to stimulate respiration. In contrast, opioids act on pacemaker neurons via mu receptors and inhibit breathing. Hence, opioid overdose can cause respiratory depression. However, it is found that serotonin acting through 5-HT4 receptors on these neurons prevents opioid-mediated respiratory depression, leaving their analgesic action intact. Okay, so we have studied that rhythmic respiration at rest is brought about by Pre-Bötzinger complex in association with DRG, whereas VRG comes into action whenever there is need for increase in rate and depth of the respiration, that is during forceful breathing. Now let's see about Pontine centers and how they modulate activity of medullary centers. We shall begin with apneustic center. Apneustic center is located in lower pons. Stimulation of apneustic center leads to stimulation of DRG, leading to prolonged inspiration with brief or insufficient expirations. This type of breathing is termed as apneusis, and hence the center causing apneusis is called as apneustic center. Normally, activity of apneustic center is inhibited by pneumotaxic center, which is another Pontine respiratory center, as well as by vagal fibers. Now coming to the last center, that is pneumotaxic center. It is located in upper pons in nucleus parabrachialis medialis and collicular fuse nucleus. Neurons in pneumotaxic center are active during inspiration as well as expiration, and its primary function is to limit the duration of inspiration to prevent excessive lung inflation. It acts as switch off signal for inspiratory ramp and thus indirectly affects respiratory rate as well. For example, strong activation of pneumotaxic center stops the ramp signal quickly, which is then followed by expiration. The new cycle begins quickly, leading to increase in rate of respiration. This may lead to rapid and shallow breathing. The exact mechanism is not known, but it is proposed that normally pneumotaxic center inhibits apneustic center and thus limits the duration of inspiration and shortens the respiratory time. Thus, we have seen that the process of respiration is purely automatic or under involuntary control, which is regulated by medullary as well as Pontine centers. So let's summarize the working of these centers to understand the clinical aspects related to them. Respiratory centers are located in medulla and pons. Medullary centers include DRG, VRG and Pre-Bötzinger complex. DRG has only eye neurons which are active at rest. VRG has both E as well as eye neurons which are activated by DRG whenever deep forceful breathing is required. Pre-Bötzinger complex acts as central pattern generator or rhythm generator. It generates ramp signals and send outputs to the muscles of respiration. Pontine centers include apneustic and pneumotaxic center. Apneustic center stimulates activity of DRG leading to apneusis, and normally it is kept under check by inhibitory signals from pneumotaxic center as well as vagus. Vagus and glossopharyngeal nerves, which transmit sensory signals from various peripheral receptors, also inhibit DRG. DRG as well as VRG modulate activity of Pre-Bötzinger complex to regulate rate and depth of the respiration. Now let's understand the clinical aspects associated with respiratory centers. First, understand this diagram. This is the brainstem showing respiratory centers. The neighboring panel is for the respiratory pattern. The pattern in the red color indicates when the vagal innervation is intact, and the blue line indicates when the vagal influence on the respiratory center is also lost, that is due to vagotomy. The first level of lesion we discuss is below the medulla as indicated by black color line label number 1. So any injury at the caudal end of the medulla up to the second cervical segment of the spinal cord leads to respiratory arrest, and usually it is fatal because medullary centers lose their control over the respiration. Such injuries can occur due to motor vehicle accidents, stroke, tumor or neurodegenerative diseases affecting medulla, or it can be due to severe blow on the neck. So respiratory arrest is indicated as a straight line in both whether vagi are intact or their influence is absent. The second condition is where brainstem is sectioned at the pontomedullary level. Since the Pontine respiratory centers regulate the rhythmicity of respiration by modulating the medullary centers, their removal leads to an uncoordinated irregular breathing pattern. Patients may exhibit Cheyne-Stokes breathing, which involves cyclic periods of apnea and hyperventilation. Such injuries can occur due to brainstem trauma or stroke. The next lesion is between pneumotaxic center and apneustic center as represented by line number 3. This type of injury removes control of pneumotaxic center over the apneustic center, leading to its overactivity. It results in apneustic breathing, where inspiration is prolonged or gasping type with short and inadequate expirations. But if the vagus nerves are intact, Herring-Brewer inflation reflex can help to terminate inspiration to some degree. but it usually cannot fully override the excessive inspiratory drive from the disinhibited apneustic center. The last level of lesion is above the pons, as indicated by line number 4. In this case, both the medullary as well as Pontine centers work normally, and hence breathing pattern is normal. In such lesions, if vagi are cut, still the breathing pattern remains normal, but the depth of the respiration increases due to loss of sensory feedback and the reflex control. Another important clinical aspect is opioid overdose, which can lead to respiratory depression by acting through mu receptors. Since HT4 receptors block this action, treatment with 5-HT4 agonist along with opioid-based analgesics prevent respiratory depression without affecting analgesic properties. Now let's see an interesting condition, Ondine curse. The name Ondine curse originates from an European myth about Ondine, a water nymph or mermaid-like spirit. According to the legend, Ondine fell in love with a mortal man and gave up her immortality to be with him. However, he betrayed her by being unfaithful. Heartbroken, Ondine cursed him, saying, "As long as you are awake, you may breathe, but should you ever fall asleep, your breath will abandon you." As a result, he could no longer breathe automatically during sleep and eventually died. Thus, in this condition, person loses the involuntary control of respiration and hence can breathe normally by voluntary efforts while awake, but may stop breathing during sleep and require ventilatory support. In medicine, rarely similar condition is seen due to severe injury or trauma to brain or brainstem and called as central hypoventilation syndrome. It may be congenital or acquired, and a person loses automatic control of breathing. This poetic yet tragic myth highlights devastating consequences of losing involuntary control over breathing. So let's conclude the session by summarization. Respiratory centers in medulla and pons regulate rate, rhythm and depth of the breathing with inputs from various receptors. Any damage or dysfunction in these centers can lead to serious respiratory abnormalities. Understanding the role of respiratory centers is crucial in clinical practice, especially in critical care and neurology. So that's all for today. Thank you for watching and see you in the next video. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.